So you might be wondering, how do we just go from DNA to this blue and pink walnut-shaped adenodeaminase protein that is so crucial for good immune function? Well, first of all, the actual adenodeaminase protein doesn't actually look blue and pink and walnut-shaped inside of our bodies. This is just another way of showing you the different chemical interactions that might happen inside the protein. Now, the way that this protein is shown here is its tertiary structure. What is the tertiary structure? It's the form of the protein that can finally perform its function. Now, let's have a look into what really goes on in the protein making factories and how we can go through the primary, secondary structures to finally reach the tertiary structure. Well, the first thing that happens is that this DNA is made up of 4 molecules, and those 4 molecules get red. The 4 molecules are adenine, thymine, guanine, and cytosine. So these 4 molecules get red and they basically get rewritten. And as they're getting rewritten, this thymine kind of gets dropped out. It gets replaced with a uracil instead. And this next sequence is known as mRNA, also known as messenger RNA. Now this sequence, it's very short lasting and it gets quickly sent off to the factories. These factories are also known as ribosomes. And as this sequence passes along the factory's belt, we have these powerful machines known as transfer RNA, which read the sequence as it's getting passed along. And what they do is they attach the complementary base onto the sequence that is passing along. So A is complementary to U, vice versa, and G is complementary to C, vice versa. And these sequences, they get read 3 letters at a time. And every time we find complementary bases for these 3 letters, we produce an amino acid. And as each amino acid is being produced, they fit into this growing chain. So these are just labels that we've given. What do they kind of look like on a chemical level? Well, we have a bunch of these atoms, and depending on what the R group is, whatever molecule is there, we have a different amino acid. So this amino acid could be different to this one, and this one, and this one, and so on. Now, once we've got this really long chain, it will be very long. And depending on the R group, we identify what the amino acids are. We also have to remember that chemicals are constantly moving around inside of our cells. And depending on the chemical properties, this R group might be scared of this R group and want to stay away from it. Whereas this R group might actually want to be friends with this R group over here and might want to move closer to it. So this is a primary structure. And depending on whether the R groups want to be friends or enemies, we get the secondary structures. So there are 2 main secondary structures. There are the alpha helices, which are curly looking shapes, and there are the beta pleated sheets, which look like folded paper. When we look into aminodeaminase in a bit more detail, the alpha helices will be shown as how they look here. However, the beta pleated sheets will be shown as arrows.